Health Presentation 3. Why are enzymes important? Enzymes aid cell metabolism by acting as catalysts. That is, they help the cell to do its work of converting nutrients into energy the body can use. I cannot emphasize enough the importance of enzymes because education in this area has been largely neglected. Enzymes are produced by ribosomes of plant cells, which are in the cytoplasm, that is, the part of the cell that is outside the cell nucleus. After an enzyme is synthesized, it will be altered so that it can be used in our own cells. A substance that speeds up a chemical reaction without being a reactant itself is called a catalyst. The catalysts for biochemical reactions that happen in living organisms are called enzymes. Enzymes are usually proteins. Enzymes perform the critical task of lowering a reaction's activation energy, that is, the amount of energy that must be put in for the reaction to begin. For example, think of starting an engine by turning the ignition key. Enzymes bind themselves to certain molecules and hold their forms in such a way that chemical bond breaking and bond forming processes take place more readily. Diagram A is a reaction coordinate diagram showing the course of a reaction with and without a catalyst. With the catalyst, the activation energy is lower than without the catalyst. Notice that the catalyst does not affect the delta G, that is the change in energy for the reaction. To clarify one important point, enzymes do not change a reaction's value. That is, they don't change whether a reaction is energy releasing or energy absorbing overall. That is because enzymes do not affect the free energy of the reactants or products. Instead, enzymes lower the energy of the transition state, an unstable state that products must pass through in order to become reactants. The transition state is at the top of the energy hill in the diagram above. In other words, enzymes make it easier for the production of energy within the cell. It's easier for them to climb the hill. What are active sites and substrates? To catalyze a reaction, an enzyme will grab on or bind to one or more reactant molecules. These molecules are the enzyme's substrates. In some reactions, one substrate is broken down into multiple products. In other reactions, two substrates come together to create one larger molecule or to swap pieces. In fact, whatever type of biological reaction you can think of, there is probably an enzyme to speed it up. The part of the enzyme where the substrate binds is called the active site, since that is where the catalytic action happens. As a substrate enters the active site of the enzyme, it helps form the enzyme-substrate complex. The reaction then occurs, converting the substrate into products and forming an enzyme products complex. The products then leave the active site of the enzyme. Proteins are made of units called amino acids. In enzymes that are proteins, the active site gets its properties from the amino acids it is built out of. These amino acids may have side chains that are large or small, acidic or basic, water absorbing or water repelling. The set of amino acids found in the active site, along with their positions in three-dimensional space, give the active site a very specific size, shape, and chemical behavior. Thanks to these amino acids, an enzyme's active site is uniquely suited to bind to a particular target, that is the enzyme's substrate or substrates, and help it undergo a chemical reaction. What are environmental effects on enzyme function? Because active sites are finely tuned to help a chemical reaction happen, they can be very sensitive to changes in the enzyme's environment. Factors that may affect the active site and enzyme function include temperature. A higher temperature generally makes for higher rates of reaction whether enzymes are present or not. However, either increasing or decreasing the temperature outside of a specific range can affect chemical bonds in the active site, making them less likely to bind substrates. Very high temperatures may cause an enzyme to denature, losing its shape and activity. In humans, temperatures of 37 to 40 degrees Celsius will denature enzymes. Temperatures above 40 degrees Celsius will destroy enzymes. Active site amino acid residues often have acidic or basic properties that are important for catalytic action. Changes in pH can affect these residues and make it hard for substrates to bind. Enzymes work best within a certain pH range, and, as with temperature, extreme pH values, either acidic or basic, can make enzymes denature. How are enzymes made to bind to a molecule's substrate? The matching between an enzyme's active site and the substrate is not just like two puzzle pieces fitting together. Instead, an enzyme changes shape slightly when it binds its substrate, resulting in a very tight fit. This adjustment of the enzyme to properly fit the substrate is called induced fit. When an enzyme binds to its substrate, we know it lowers the activation energy of the reaction, allowing it to happen more easily. But what does the enzyme actually do to the substrate to reduce the energy needed for activation? The answer depends on the enzyme. Some enzymes speed up chemical reactions by bringing two substrates together in the right orientation. Others create an environment inside the active site that is favorable to the reaction, for instance, one that is slightly acidic or nonpolar. The enzyme-substrate complex can also lower activation energy by bending substrate molecules in a way that facilitates bond breaking, helping to reach the transition state. Finally, some enzymes lower activation energies by taking part in the chemical reactions themselves. That is, active site residues may form temporary covalent bonds with substrate molecules as part of the reaction process. An important word here is temporary. In all cases, the enzyme will return to its original state at the end of the reaction. It won't stay bound to the reacting molecules. In fact, a hallmark property of enzymes is that they are not altered by the reactions they catalyze. When an enzyme has finished catalyzing a reaction, it just releases the product or products and is ready for the next cycle of catalysis. The following video, How Enzymes Work, will illustrate how each of the elements we have just discussed work in harmony to help the cell accomplish its purpose.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.